Another day, another story. Scientists may have uncovered a link between the risk of autism in children and fatty acids in umbilical cord blood, but one Harvard professor told Newsweek more work needs to be done in the area. Recent studies have provided significant insights into the causes of autism. Researchers at UCLA have linked genetic risk factors for autism to specific cellular changes in the brain. Welcome to Tabo Eminent Channel. By analyzing over 800,000 brain cell nuclei, they identified key neuronal and glial cell types affected in autism. They also pinpointed transcription factor networks that regulate gene expression in these cells, suggesting a direct connection between genetic factors and brain changes in autism. These findings could lead to new therapeutic approaches for autism spectrum disorder. The study, led by UCLA neurogeneticist Dr. Daniel Geschwind, is part of the National Institutes of Health Psych ENCODE project, which maps gene regulation in the brain. They discovered that specific neuron types and glial cells show the most profound changes in autism. The study identified transcription factor networks that drive these changes, linking genetic risk factors to observed brain alterations. The researchers found that both neurons and glial cells exhibit significant changes in autism, particularly neurons that connect brain hemispheres and somatostatin into neurons. They identified specific transcription factor networks that regulate gene expression in these cells, linking genetic risk factors to brain alterations. This research could pave the way for new therapeutic strategies for autism spectrum disorder. To shed light on the possible causes of autism, researchers from the University of Fukui investigated the link between polyunsaturated fatty acids, PUFA, in umbilical cord blood samples and autism scores in 200 children. They believe that this newfound knowledge could be crucial for understanding, diagnosing, and even preventing autism spectrum disorder, ASD. The study identified one particular compound in the umbilical cord blood acid, called diheta, that may have strong implications for ASD severity. Baby umbilical cord. A baby with an exposed umbilical cord. A fascinating new study has uncovered a link between autism risk and umbilical cord fatty acids. The levels of diheta, an arachidonic acid-derived diol, in cord blood at birth significantly impacted subsequent ASD symptoms in children and were also associated with impaired adaptive functioning, Hideo Matsuzaki, study author and child mental development professor at the University of Fukui, said in a statement. These findings suggest that the dynamics of diheta during the fetal period is important in the developmental trajectory of children after birth. Specifically, the authors found that higher levels of diheta were associated with difficulties in social interactions, while low levels were linked to repetitive and restrictive behaviors in children. Notably, this correlation was more pronounced in girls than in boys. The umbilical cord samples were collected and preserved immediately after the children's birth. ASD symptoms in these same children were then assessed at 6 years old with the help of their mothers. Building on these results, the researchers suggest that measuring diheta levels at birth could soon become a valuable tool for predicting a child's risk of developing ASD. The effectiveness of early intervention for children with ASD is well established, and detecting it at birth could enhance intervention and support for children with ASD, Matsuzaki added. He also said that inhibiting diheta metabolism during pregnancy might be a promising avenue for preventing ASD traits in children, although more research will be needed in this area. Scientists want to build an ark on the moon. Here's why. Cancer study reveals Gen X, millennials at greater risk than boomers. Be cautious. US chocolate and cocoa tests reveal unsafe lead levels. The study, published in Psychiatry and Clinical Neurosciences, builds on earlier research in mouse models that suggested PUFA and their metabolites during pregnancy play a crucial role in ASD development. Sharing the motivation behind the research, Matsuzaki said, "Psych metabolism forms both epoxy fatty acids, EPFAs, which have anti-inflammatory effects, and dihydroxy fatty acids, or diols, which have inflammatory properties. We hypothesized that the dynamics of psych-PUFA metabolites during the fetal period, that is, lower EPFA levels, higher diol levels, and/or increased EPFA metabolic enzymes would influence ASD symptoms and difficulties with daily functioning in children after birth. The study authors believe these findings open a promising avenue for unraveling the mysteries of ASD, potentially leading to improved diagnostics and treatment. While he recognizes the area of research under focus in this study is emergent, James McPartland, professor of child psychiatry and psychology at Yale University School of Medicine, told Newsweek that the research does not provide information currently useful for diagnosing ASD. This is an exploratory study investigating potential mechanisms for autism clinical features that are not established and presently not well understood. Its significance lies in generating hypotheses to be tested in more rigorous future studies, but it provides limited information on which to draw conclusions or guide clinical decision-making, he said. McPartland also noted that prevention of ASD is not an objective of most autism clinicians and researchers. Instead, the primary objective is to advance research and develop insights that can enhance the quality of life for autistic individuals and their families. Of note, the Autism Diagnostic Observation Schedule used in this study is not a sufficiently sensitive and specific diagnostic tool when used in isolation. It is most reliable when combined with parent interviews and clinical judgment according to DSM-5 diagnostic criteria, he added. From the published data, it cannot be determined what proportion of the individuals studied would actually meet diagnostic criteria for autism. Dr. Daniel Geschwindt's team at UCLA used single-cell genomics to map gene regulation in the brains of individuals with autism, revealing significant changes in specific neurons and glial cells. They identified transcription factor networks that drive these changes, establishing a direct link between genetic risk factors and brain alterations. This research offers new insights into the molecular mechanisms of autism and potential therapeutic targets. In addition to the groundbreaking findings linking genetic risk factors for autism to specific brain changes, the UCLA study sheds light on how these genetic influences manifest at the cellular level. Researchers found profound alterations in neurons that connect the brain's hemispheres and in somatostatin interneurons, which are crucial for brain circuit development. The identification of specific transcription factor networks provides a deeper understanding of the molecular mechanisms driving autism. These insights could lead to new, targeted therapeutic approaches to treat autism spectrum disorder. The UCLA study is a major advancement in understanding autism. By analyzing brain tissue from individuals with autism and neurotypical controls, researchers identified significant changes in specific neuron and glial cell types. These changes were linked to transcription factor networks regulating gene expression, which are influenced by genetic risk factors for autism. The findings offer a detailed view of the molecular mechanisms driving autism and open new avenues for developing targeted treatments. This comprehensive study marks a significant step forward in autism research. Thanks for watching. Request you to subscribe the channel.